so we can talk a little bit about that when we get there.
Hi, Dr. Kevin.
Yes.
Yeah, I just found a paper wherein they were describing how and when bicupilin is used. So it is a convulsant basically, and acts on GABA A receptors, and by doing so, like it also inhibits the receptor. So yeah, like if given to an animal model, it is usually used to study anti-epileptic drugs. Given it produces convulsions,
Right.
and it's a very good model for studying anti-epileptic drugs.
Very good. Did anyone find any other uses for bicupilin?
So I found saccharine and metrazole and flumazenil.
So other drugs, yes.
Yeah, three other
And
drugs.
did you find out any other reason these drugs are used?
So when I just googled it,
benzodiazepine overdose or any GABA receptor agonist overdose can be antagonized by this drug.
Right. Is that what you found, Young?
Yeah. Yes.
Yes. Okay. So, GABA agonist overdose. So GABA agonists like barbiturates, which are not used too much anymore, but barbiturates are benzodiazepines. Very good. Okay, good. So, so the GABA receptor then acts as so GABA itself is the main inhibitory neurotransmitter in the brain, right? In the CNS, or really in the brain, because we'll talk about glycine in a minute. And how it works is that GABA is a chloride ion channel. So that means that when the GABA receptor is open, and here's the GABA receptor in the cell membrane here. When the GABA receptor is open, that means that chloride ions are going to flow through that channel into the inside of the cell, into here, right? So the first thing that you'll recognize here about the GABA receptor is that it's 5 subunits, 2 of which are alpha here, and 2 beta, and 1 gamma. And I have another sheet that I put on your your canvas, excuse me, that goes into these subunits a little bit more. So I'm going to pull that sheet up. Just give me a second here. Notability, it's easier. Right in. So this receptor, so the way you were looking at it in your handout, here, let's just go back there one more time. Here in this handout, so here's the cell membrane here, right? And then you can see the receptor. So you're looking at it like a side view. So it'd be as if you're driving by a house on a street if you do Google Maps and you do Street View and you see all the houses going by, or if you're driving by yourself, you'd be looking at the exterior of the house. And this is kind of what you're looking at here, the exterior of the receptor. But if you want to look at it a little more closely, shown here, in this view here, this is like top down. So this is like an aerial view. So again, if you're thinking about Google Maps and you put on satellite view and you see the tops of the houses, this is what you would look like. It would look like if you were looking at the receptor from the top, right? And then here would be the membrane. here, whatever, right? Okay, so it's going through the membrane. Actually, the membrane would encompass the whole thing, so I should write it correctly. The membrane would really be like out here, cell membrane, right? And then, you can see the lipid bilayer here. Okay. All right, so then what this looks like, so you can see that the alpha and the beta subunits, they alternate with each other. So you have alpha, beta, and then between the last alpha, the second alpha and beta, you have the gamma subunit here. And then there's this pore in the middle where the chloride ion can go through, right? And then you can see that GABA, which is the endogenous ligand for these receptors, it binds between these alpha, binds between the alpha and the beta subunits here. But if you have an agonist, the agonists that we use clinically, like benzodiazepines, they bind allosterically, which I think Dr. Johnson went over with you. Allosteric means it binds to a different site than the ligand. And so you can see that the benzodiazepine, for example, the binding site is between the alpha and the gamma subunit. And then each of these subunits are actually made up of 4 further divided subunits. So 4 smaller subunits make up one of these big subunits. And then, like I said, they all come together to form the GABA receptor. The reason why I wanted to include this slide is that there's not, here in this picture up top, you see alpha 1, beta 2, and gamma 2. Well, there's actually different subtypes of these subunits. So down here at the bottom, you'll see that there's alpha 1 through 6 and beta 1 through 3, and then gamma 1 through 3 too. And then there's some other receptors that we don't talk much about that are, I personally don't know as much about, and I don't think there's as much as in the literature about it. But there's instead of gamma subunit, excuse me, you could have delta, epsilon, pi, or theta. So the reason why I point this out is because the way these subunits come together help to determine their function. So if you have alpha 1 and gamma 2, like in this picture up here, those are, when GABA binds, it's mainly a sedative effect. And it inhibits neurons in a phasic manner, meaning that it causes inhibition of firing at a very regular rate. And then the other main type of GABA receptors, the GABA A receptors, still have the gamma 2 receptor subunit, but then they can also, instead of having the the alpha 1 subunit, they have alpha 2 or alpha 3. If that's the case, if they come together in this manner, these are mainly anxiolytic. They're not as much sedative. So the reason why this is important is because for drug discovery and use of GABA antagonists in order to produce anxiolytic effects, which is really sometimes the main idea. We don't want to make the person completely sedated. We'd rather target this anxiolytic type of receptor. So for drug discovery, if we could find drugs that bind more preferentially to this makeup of the GABA receptor, then we could reduce the sedative effects, which a lot of patients don't want if they just want the anxiolytic effects, and increase those anxiolytic effects. Now, sometimes we do want sedation, so we would obviously target more this receptor that's made up of the gamma 2 and alpha 1 subunits. So I just wanted to point that out, that it's not all just depending on, it's not just one homogeneous receptor, I guess. Not all, back up. Not all the GABA receptors are exactly the same. They're going to be different depending on what subtype of subunits make up the receptor. Okay.
Ma'am,
Yes.
I have a question.
Sure. Who is this Musa?
Yeah, ma'am. I heard that, like, I read it about the GABA receptors before, and the binding site, is it that the binding site of the benzodiazepine is similar in all conditions, or is there are different binding sites of the benzodiazepines? Like, I've heard that there are two binding sites, that it's BZ1 and BZ2,
Oh, okay.
or is it like these are the different subtypes of the GABA receptors? Like, what's the difference?
Yeah, I mean, honestly, Muslim, I don't know. I'd have to look that up. So you've read that there's benzodiazepine site binding site 1 and binding site 2?
Yeah, ma'am. Like,
Okay.
I read it, like you told us the type of different types of the GABA receptors. Like, I have read in some place, I can show you the reference as well.
Okay.
That maybe that there are different types of benzodiazepine receptors, like BZ1 and BZ2. And
Okay.
when drug binds to BZ1, it produces sedative and phasic effect, as you have shown. And when drug binds to BZ2, and it produces the anxiolytic effect.
Yeah. So that sounds like it has to do with those subtype of subunits, right? So
Maybe.
yeah, that's what it sounds like. I don't know that the binding site is different. I think the binding site stays here, like I showed here, between the alpha and gamma, but instead for the anxiolytic effects, it would be alpha 2 here, right? And gamma 2, same there. I don't know that the binding site is actually different, but I'd be interested in looking at those references because I don't know that literature.
Yeah. And just one more question, ma'am. Like,
Sure.
are the
Yeah.
barbiturates also bind to the same position
Yeah.
where the benzodiazepine binds?
Yes, they bind to the same site.
Okay, ma'am. Thank you.
Mm-hmm. Good question. Yeah, send me that reference. I'm interested in learning about that or reading about that. Okay, so like I said, it's the major inhibitory neurotransmitter in the CNS. And what it does is it decreases neuronal firing rate and hyperpolarizes cell membranes. So I don't know how much Dr. Johnson, did he go through action potentials and such?
Yes, ma'am.
Oh, he did. Okay. So then he probably told you that in order for a neuron to fire an action potential, right, there's they have to reach what is called threshold, right?
Yes.
And when we talk about it, we just talk in a very general case. This is not going to apply. It applies to most neurons, but some neurons have a little bit of different membrane voltage. So across the neuronal membrane, right, if you have the cell membrane here, there's a charge across here.
Mm-hmm.
And it's a negative, and when the neurons are resting, it's a negative number. And it's really a misnomer. Neurons never really rest, right? But we know experimentally from taking them out of the brain of rodents and putting them in a dish, we can get them to semi-rest. And we can measure this voltage across their membrane, or this charge. And inside the neurons, there's a lot of negative charge here, right? Negative charge is higher. And outside the neuron, the positive charge is higher. But because the inside is so negative relative to the outside, we call the resting membrane potential of a neuron a negative number. We call it negative 70 as a general case. It could be a little bit different from, in some neurons, but as a general case, we call this negative 70. And then I guess Dr. Johnson told you, there needs to be some kind of depolarization of this membrane up to this threshold value here. Does this sound familiar? Is this the stuff you went through?
Yes.
Okay. So up to this threshold here, just repeat it for a second. And then you get the actin potential with a large depolarization, and you get a repolarization, a hyperpolarization, and then back to membrane potential. So this is polarization. And then this is repolarization. So, and then repolarization. So that should be somewhat a review. So the whole getting to this threshold here depends on the influx of positive ions into the neuron. So you need an influx or a net influx of positive ions. So if the neuron on its membrane then has the GABA receptors, right? So we'll just draw these little GABA receptors. And it starts at this minus 70. What's going to happen is when GABA binds, and you get this influx of chloride ions, instead of going positive towards threshold, you're going to get it going negative down away from threshold, right? So GABA is going to cause what we call hyperpolarization. And it's going to make that neuron, it's going to make it harder for that neuron, rather, to reach this threshold up here at negative 55. Right? So that's what happens when GABA binds. It hyperpolarizes the cell membrane. It makes it much harder to reach this threshold. This gap is much larger. So you need a much larger influx of some kind of positive charge in order for an action potential to occur and neuronal firing to occur. So in effect, what it does then is it decreases the neuronal firing rate. And GABA receptors are found all throughout the CNS. I actually have a picture. Let me see if I can find it from another class to show you exactly what I mean by it being everywhere. Let's see if I can find these notes. And I can post this for you. It's from a different class, just to show you the distribution. It's from my HPP class to the PharmD students. So Matt, this might be familiar to you once I pull it up, once I can find it. Neurotransmitters. There we go. Okay, so just to show you where GABA is in the brain. Here's the GABA distribution. Okay, so it's produced in these nuclei here in like the so near the midbrain, and then you can see, and it's also produced in some nuclei over here in the cortex and in the cerebellum. And you can see how this distribution goes all throughout, right? The frontal cortex, all throughout the limbic system here, not as much in the brainstem, but then there's a lot also in the cerebellum. So it's all throughout the brain, and it makes sense because it is, the main inhibitory neurotransmitter in the brain, and it helps regulate neuronal firing. Without it, cells would wear out, the neurons would wear out because they'd be constantly firing, and we get excitotoxicity. Okay, and then we know the agonists are, oh, here they are. They're coming up. We know that you looked up this antagonist, and by bicuculin and others, and you've already told the class that it's a convulsant, and it acts on the GABA A receptor and can be used to study anti-epileptic drugs. But it's also useful if there's an overdose of GABA agonists. So the GABA agonists listed here are barbiturates, ethanol found in obviously in alcohol that is a GABA agonist and can slow neuronal firing. Benzodiazepines and propofol. Propofol is used in anesthetic pre-surgery anesthetized to help with during anesthesia. Excuse me. Barbiturates have fallen out of favor. They're not used as much because they were highly abused, and they're such strong GABA agonists. And so we already talked about the effects it can have. It can have anxiolytic effects, sedative effects, sedative and hypnotic effects. We often put those together. They can be used as muscle relaxants and can cause anticonvulsant. They're often used in epilepsy to treat the convulsions that epileptic patients have, and also can cause some changes in memory. They can cause amnesia. And that's because GABA and glutamate are both really important in what's called long-term potentiation, or the formation, which is involved in the formation of memories. So the counterpart, or the similar neurotransmitter that's in the CNS but is more constricted to the brainstem and the spinal cord, is glycine. It binds, instead of binding GABA, it binds the inhibitory neurotransmitter glycine, but it works very similar to the glycine and glycine receptor work very similar to GABA in that they're chloride ion channels. So it's going to increase influx of chloride. And also cause hyperpolarization and decrease neuronal firing. And actually, glycine is, when it's released in the central nervous system, it actually will also, in the spinal cord, when it's released, it will decrease skeletal muscle contraction, because it's those in the spinal cord. There's lower motor neurons there that have glycine receptors on them and glutamate receptors on them. So if you have this lower motor neuron, in the spinal cord, here's the cell body, and it sends out signals to the skeletal muscle. These neurons release acetylcholine. Oops, I'll just put ACH. Acetylcholine, and then that causes skeletal muscle contraction. Okay, so that's only if glutamate binds, because glutamate is the excitatory neurotransmitter. It's going to activate these neurons, and it's going to cause the release of acetylcholine and skeletal muscle contraction. On the other hand, if you have glycine released in the spinal cord, it binds to those glycine receptors on the lower motor neuron. It'll inhibit this action potential, and it'll cause muscle relaxation. So two of the agonists here are glycine and taurine, that will inhibit muscle contraction. And then these two are poisons that will actually cause spastic paralysis, which means you'll get over stimulation of the skeletal muscles. Okay. So next we're going to go on to kinase receptors. So kinase receptors are so far I don't know, he was talking about so we talked about the ion channels, GPCRs, right? So you did G protein coupled receptors. Now we just talked about a few ion channel receptors there, are ionotropic receptors, and now we're going to talk about kinase receptors. So kinase receptors are receptors usually for hormones or growth factors. I like to call them growth factors, but the growth factors are indeed hormones. So did you cover the definition of what a hormone would be? Would someone want to tell me what's special about hormones?
When they're secreted from one area and their target effect is somewhere else.
Yeah, so they're released from one area, usually somewhere in the brain, the pituitary gland usually, or for insulin from like beta cells, right? So in the pancreas. So what's special about them though? What's different from them than, say, I don't know, let me just say like a neurotransmitter that's, released in the brain? Where do these get released into?
They are released into the blood.
Right. So what does that mean in terms of their action?
Their action is far away from the releasing site.
Yes, right. So they can have widespread effects, right?
Yeah.
However, they're not going to affect everywhere. So say, for example, a growth factor or another hormone, like let's just take estrogen. Estrogen's released into the bloodstream.
Yep.
Is it going to affect every single tissue? What needs to be present in order for that? Or estrogen's not a good example because it doesn't use well, let me think of another one. Let's just use one of these growth factors released into the bloodstream. Is it going to affect every tissue equally?
No.
No.
Yeah, I think it will depend on the presence of the receptors.
Exactly. Yep. Good. Very good. Okay. So these are different than the receptors we've studied before, the G protein coupled receptors and the ionotropic receptors, because they exist in the membrane as a single peptide chain. So there's a picture down here. So they exist as just this single one peptide chain. And when I draw them in class, I often use, here's the membrane, and then I'll draw this. And it exists like this as a single peptide, just transmembrane, just spanning the membrane one time, right? And inside is a kinase domain on this receptor. And then there's binding domains down here for the intercellular components that need to bind to the receptor. And then this is the extracellular fluid, obviously, and this is the intracellular fluid. And then here would be where the ligand would bind, right? Okay. But before the ligand binds, so let's undo that for a second, they exist as a single peptide chain or a monomer. It's a monomer. And when they're in the monomer state, they are inactive. Okay? So right now, this receptor is inactive, but maybe down here on the membrane a little bit further, there's another one of these receptors hanging out, okay? All right, so what happens then when the hormone binds or the ligand binds? Here, it causes these receptors to come together and form dimers. So now in the membrane, you'd have these two receptors very close to each other. So what that triggers then is these kinase domains down here in the intracellular part. That triggers them to become active, and it causes an autophosphorylation of these receptors. So you get a couple phosphate groups put on here. And causes this autophosphorylation. So this is then the signal for adapter molecules to bind. And the main adapter molecule for these is called GRB2. And so let's just go down and look at our figure here. So here's the dimer. Here's the adapter molecule, GRB2. And another adapter or cofactor that comes with it is this sauce. It's recruited along with GRB2 to the receptor. And then from there, what happens is you get activation of the RAS MAP kinase cascade. The RAS in this case here is acting like a G protein. It is a G protein, but it's it only has one. It doesn't have 3 subunits like the classical G proteins. It's a single subunit. G protein. But it works exactly the same in that GDP is exchanged for GTP when it's activated. And like the G proteins, they often go on to eventually activate signaling cascades. The same thing with RAS. RAS goes on to activate intracellular signaling cascades, and specifically it often activates these MAP kinase cascades here. And did Dr. Johnson go over MAP kinase signaling at all?
No.
No. Okay. So what happens with MAP kinase signaling is, so we're going to be inside the cell now, and we have, in this case, it's RAS MAP kinase, but other things can activate MAP kinases, but we'll just use the RAS. We have the GDP exchanged for GTP, sorry, the other way around. GTP coming on and kicking off the GDP. This is then going to recruit what we call, actually, let me back up. MAP kinases, that equals mitogen, protein kinases. I'm sure through your research seminars and such, most of you have heard of these before, because my lab and others, we work on these MAP kinases. And so once the RAS is activated, it'll activate what we call a MAP kinase. So all that means is it's it's 3 upstream from the MAP kinase, and the reason why it's named like this is it goes backwards up from the MAP kinase. So let me just get through the whole pathway, and I think you'll understand. So that MAP kinase, in this case, is called RAF. There's other MAP3Ks, we call that sometimes too. And then once that becomes activated, it gets phosphorylated. So all of these kinases get phosphorylated in turn. And then downstream from RAF is the MAP 3K is a MAP2K, MAP kinase, and we often call them MECs. That's the abbreviation. And then finally, you get to a MAP kinase, and the most studied MAP kinases are ERKs, which stands for equals extracellular signal regulated kinase. And there are at least 7 or 8 of those. So there's like ERK 1 through 7 or 8. Lost count. My lab looks specifically at ERK 1, 2, and ERK 5. ERK 1, 2 are often studied together because they have they often are activated together. Phosphorylation here, and these become phosphorylated too. There's also other MAP kinases. One known as P38, that's just simply protein 38. And then there's also one called JNK, and that's JNK stands for C-Jun kinase. And we often call that junk. All right. So these are the three, ERKs, P38, and junks are the main MAP kinases. And then what these can go on is then do all these kind of cellular functions. Can cause cell proliferation, can cause inhibition or apoptosis. In the case of P38 and junk, they actually are more commonly known to increase apoptosis than inhibit apoptosis, whereas the ERKs more commonly inhibit apoptosis and promote cell proliferation. So these pathways are activated downstream of these kinase-linked receptors. So just back to the naming for a second. So the MAP kinases are down here at the end of the chain. And then what it kind of was like found in reverse. So when they found the upstream component of the MAP kinase, they called it a MAP kinase, yes, because it is a kinase. So kinases are enzymes that phosphorylate other things in the cell, other proteins in the cell. So it is a kinase for MAP kinase, right? So that's where we get the name MAP kinase. And then up here, if you the line here, this RAF is a kinase for a MAP kinase, which is MEC. Okay, so the naming gets a little confusing and it gets a little redundant. So not a lot of people talk about it in that way anymore. They usually just name, if they're talking about a certain cellular signaling pathway, they'll actually use the names of the protein. So in this case, we would say RAS goes to RAF, that goes to MEC, that then in the figure goes to ERK. Okay, we don't say MAP kinase. It just gets over redundant and people get lost. But I just wanted you to see where those names came from. Okay, so back to this receptor. So is there any questions about that signaling pathway? I mean, this is a very simplified version. It actually gets quite complicated inside a cell. It was once thought that these pathways were parallel to each other. So so in other words, let me just insert a page here. It used to be thought that, okay, I'm not going to put, I'll put the cell membrane here just to be complete. So then we have mitogens out here. Mitogens mean like things that cause cell growth, can be the growth factors. It can be stress can activate these receptors. But anyway, so we have these kinase-linked receptors here activated. Right, and it used to be thought, and I'm just going to do downstream. It used to be thought it was so like here, RAF to MEC to ERK, and then you have over here, it's not, it would just be KK, to MEK to P38. And then over here, we'd have a different MEKK to MEK to Jun kinase. So when they were first discovered, actually my postdoctoral advisor, she was one of the first to show that these ERKs are mainly cell survival, and these two are mainly cell death. And when she started studying them, this was back in the 1990s, late 80s, 90s. When she started studying them, it was thought that these were 3 parallel pathways. We now subsequently know that there's this kind of stuff going on, all this kind of crosstalk, right? Those might not be exactly the way the arrows go, but there's crosstalk all over the cell. So it's not just a simple 3-tiered pathway anymore. It's all this different kind of crosstalk. And it causes a lot of redundancy in signaling in the cells. And that becomes an issue when you're trying to block, say you're just trying to block this pathway here. Well, a lot of times what can happen is it can circumvent that by having this other MEK over here activate this ERK. So a lot of times when we're using inhibitors of these pathways, we'll see a compensatory upregulation of another pathway. And that's particularly true within the ERKs, like ERK 1 and 2. We've seen that if you inhibit that pathway, you get an upregulation of our favorite pathway, ERK 5. We've also seen that if you block these pathways, these ERK pathways, sometimes you get an upregulation of other kinase signaling pathways that are not necessarily considered part of the MAP kinase family, but they're also important for cell survival. And that's shown back here on your this diagram, specifically this phosphatidylnositol-3 kinase pathway can be compensatorily upregulated, at least we found in the lab, if you block these ERK. Okay, so that's a very quick overview and a very simplified way of looking at the MAP kinases. If you were to look in the literature, there's so much literature on it. It would take more than a lifetime to absorb everything that happens. But I thought I'd just give you a little bit of background on these cascades. Any questions about that?
Yeah, Dr. Kavanaugh.
Yeah.
I'm not quite sure about the function of the adapter that you mentioned before, the RAS-RAF.
Oh, yeah, the
GLP-1.
adapters. Right. So the function of that is just to recruit the RAS to the membrane and also to activate the RAS.
Okay.
Okay. Yeah. So it's just like recruiting all these things to this receptor in order to get it activated.
Ma'am,
Yes.
I want to know that when a ligand binds to this tyrosine kinase receptor, ma'am, is it that it will always activate a GRB2 system, and if GRB2 system is inhibited, then it will go for the compensation one, or it can start any of the pattern, like it can start with PI3K or STAT or GLP2.
Yeah, no, so right. So you can start, these receptors also activate directly PI3 kinase pathway. That's right. And there are other adapter proteins, but these are the most common, and these are the ones that are used for this RAS, RAF, MEK, or pathway.
So how it depends, that it will start PI3K, STAT, or GRB2?
How does it decide? That's a good question. Yeah, it's a good question. Usually, in my experience in the lab and also things I've read, if a growth factor binds these receptors, these pathways will both get simultaneously activated.
That's great.
Okay, so simultaneously.
As long as all the components are there in the cell, these pathways are gonna become activated.
Mm-hmm.
Okay, thank you.
Yeah. Good questions. Mm-hmm. Okay. So, and then the way that these cascades promote cell survival is that they eventually there's downstream, so then there's targets of these. So really, I should not have drawn that arrow right to that, because that's not, well, that's, yeah. So there's targets of these proteins. And often these targets are transcription factors. Or repressors or activators of transcription. And what happens with these targets is these targets get translocated to the nucleus. Sometimes the MAP kinase actually itself gets translocated to the nucleus along with its target. Sometimes it's just the target that gets translocated to the nucleus. And once these targets get into the nucleus, then they interact with other transcription factors, repressors, or activators of transcription. So there's like that, it goes to this. And then eventually you get transcription, gene transcription of proteins that are going to affect cell proliferation, apoptosis, migration, cell cycle progression, things like that. So, so then you get activation of gene transcription factors, or like I said, or repressors, or activators. And then this is cell proliferation because in his handout, he's focusing on this RAF pathway, but it could also be apoptosis, depending on the MAP kinase that's activated, or depending on the kinase, the transcription factor or repressor or activator, whichever is activated. You can have migration. What else? Adhesion, invasion, angiogenesis, etc. Yeah, like in the next figure. Okay, so one clinical case that Dr. Johnson is pointing out here in his notes is in some breast cancers, not all, there's overexpression of this human epidermal growth factor receptor 2, or we call it HER2. So what happens when a woman or man is diagnosed with breast cancers, they take a biopsy and then they look for the presence of receptors in the breast tissue. The receptors that they're looking for specifically are the estrogen receptor, progesterone receptor, and this HER2 receptor. So there's different types, like I said, of breast cancer. They can be estrogen, or we call this ER positive, or they can be PR positive. They can be ERP and PR positive. They can be HER2 positive, right? You can imagine all the different combinations. They can be ER, PR, and HER2 positive. And all of this, and then some of them actually don't have very high levels of these receptors, so we call them triple negatives. They're negative for all these receptors. And depending on that hormone profile, we can then say for that breast cancer, we can determine how aggressive it's going to be. So the most aggressive breast cancers are called triple negative breast cancers. That means they don't have a large expression of any of these receptors. So in that case, that means that breast tissue has escaped the need for any kind of extracellular signal in order to activate these pathways that promote proliferation. So these pathways are mutated in such a way that they are just constantly active, and the cancer is proliferating very, very quickly. Okay, so that's why those are the most aggressive types of breast cancer. They're the ones with the worst prognosis. There's also the ones that we don't really have targeted therapies for yet. It's the main treatment for them are surgery and then chemotherapeutics. And chemotherapeutics are very non-specific, right? They attack any cell that's proliferating because they're attacking those microtubules that are changing in cell division. And by doing that, any cell that is dividing in our body can be attacked and killed by these chemotherapeutics. That's why people undergoing chemotherapy for any type of cancer, right? That's why they get very sick, because the chemotherapeutics will affect any cell that's dividing in the body. So those are very dangerous and very poor prognosis breast cancers. Now, if we find the presence of one of these receptors in the breast cancer, that's a better prognosis, because that means then that we have therapies that we can target to these receptors. HER2 positive ones are a little more aggressive than either the ER and PR positive. So if you have HER2 receptors, it does cause them to be a little bit more aggressive because they're using these growth factors, not just estrogen and progesterone, to proliferate. So the growth factors, since they're pretty ubiquitous and there's a lot in the body, that they also divide fairly quickly. So if you were looking at a scale for breast cancers, the best prognosis would be like ER, PR positive without HER2 expression. Then in the middle, we'd have ER, PR positive with HER2 expression. And then with the worst prognosis would be the triple negative that don't have these receptors. And so these cancers take advantage of this signaling that we just talked about here, where the growth factor binds, activates, these PI3K pathways and these ERK pathways in order to promote cell proliferation. I think most of you know on here that I was diagnosed with breast cancer in March, and I'm very lucky because my prognosis is very good. I have ER positive, PR positive, but HER2 negative, and they found it very early, stage 1A. So I don't need chemotherapy. It was surgery, and then I will eventually need radiation, and then some medication. But I have a very good prognosis, so I was very lucky. But others, unfortunately, are not as lucky when they're diagnosed with breast cancer. So anyway, if there are HER2 receptors present, one of the therapies is called Herceptin. Let's see here.
Receptive.
Oops.
Trastuzumab.
And Herceptin is a monoclonal antibody. It's going to trigger these receptors, so it's going to bind. Oh, let me do that in another color. Oh, let me choose pink for breast cancer. Pink, it binds to these receptors. This is antibody. That's how we draw antibodies, right? I'm assuming you've seen that before. Antibody is going to bind to these receptors. And then what it does is it triggers the body to have an immune response to these antibodies that are bound to the receptor and to target these receptors to get rid of them. So it leads to these receptors being internalized into the cells, and then what happens is they fuse with the lysosomes, and the lysosomes can chew them up and downregulate them, or just leave them inside the cell where they're not accessible to the growth factor. So you get a downregulation of these receptors by using that therapeutic Herceptin antibody. Okay. Let's see what else. Where are we going? Oh, okay. Okay. All right. So then next, we're talking about cytokine receptors. Cytokine receptors are different than the receptors we just talked about, is because they have no intrinsic enzymatic activity, right? Those kinase receptors we just talked about, they have that enzymatic activity where they phosphorylate each other. And the ligands that bind to them include some growth hormones, tumor necrosis factor, interferon beta, interleukins. Right? So interferon beta is a therapeutic for MS. So it's used. These receptors are triggered in that disease. And then we have and also these receptors are kind of flared up in autoimmune diseases. There's a over activation of them especially with tumor necrosis factor. So autoimmune diseases such as rheumatoid arthritis and the such. So there's antagonists that are used, such as Humira and Enbrel, to help those patients that have the rheumatoid arthritis. Okay, so in just like the kinase receptors, in their inactive state, these receptors are also monomers, and they're going to be linked kinases that are inactive, such as this JAK kinase or this TYK2 kinase. So they have kinases on them, but as monomers, they're inactive. Okay? So again, what happens is the ligand binds and the receptor dimerizes and then activates these kinases. But instead of an autophosphorylation, this is a cross-phosphorylation. Okay, so here's this cytokine coming to the cytokine receptor, causes this dimerization, right? And this JAK will then, he has it drawn here in this figure that it phosphorylates its own. But there's really like it phosphorylates itself and also kind of cross-phosphorylates the other receptor. So there's a cross-phosphorylation of the kinases and the receptor. Okay. And then once this is phosphorylated and dimerized, it recruits this other protein. So that's the signal for the recruiting of this signal transducer and activator of transcription, or STAT, and specifically STAT2. So if we look down here, right, we have these phosphorylated receptors, and then that recruits this STAT protein to the receptors, which becomes further phosphorylated by the JAK protein. So the phospho group on the receptor recruits the STAT, but then the STAT is further phosphorylated by the JAK kinase. And then what that causes is these STATs come off the receptor and they dimerize. And then this dimer here is translocated to the nucleus. And this dimer will bind to a DNA response element, which will then affect gene transcription to generate proteins. Okay. So JAK-STAT, the cytokine receptors are really involved in an immune response, triggers immune responses. Okay, so so we have the kinase link receptors, the cytokine receptors that are a little different, and then finally, this is the final one he talks about, yes, are nuclear receptors. So nuclear receptors either reside on the nuclear membrane or they reside on in the cytoplasm. So for steroid hormones such as estrogen, progesterone, testosterone, etc., they are in the cytoplasm. And so what happens then is the hormone will bind to the receptor in the cytoplasm. From there, the receptor will go be activated. Here, binds the steroid receptor. The receptor dimerizes. I'm sorry, again, just like we saw with the other two. So the kinase linked receptors, cytokine receptors, and nuclear receptors all depend on that dimerizing. And then the receptor dimer complex actually, let's see how he has this. Yeah, the receptor dimer complex here. So here's this complex with HSP before the hormone binds. Hormone binds, kicks off this HSP, and you get the hormone and receptor complex dimerizing here. The hormone and the receptor dimer goes into the nucleus through a nuclear pore, and then binds to a hormone responsive element on the DNA, and will recruit co-activators and RNA polymerase to the DNA, which then induces transcription. So you get the mRNA transcribed. mRNA can then go back out into the cytoplasm where it's binds to the ribosomes, or is processed through the ribosomes, not really binds, it's like threaded through the needle through a hole in the ribosomes. Protein is made, and then you can have a change in cell function. So this just receptor dimer complex binds to and activates hormone responsive element binding protein, or I've also seen it just HRE here, hormone responsive element, at which then recruits the RNA polymerase and the co-activators to transcribe the mRNA and then eventually cause protein. If you're looking at the structure, organization of these nuclear receptors, they have an N-terminal domain here, then this DNA binding domain that once they get into the nucleus, but that's the part that will bind to the DNA, and the ligand binding domain here, which binds obviously the ligand, the hormone. Okay, so we have the ligands as estrogen, progesterone, etc. Antagonists such as tamoxifen and mifepristone. These are sometimes used. This is used actually to treat those ER positive breast cancers. And then for non-steroidal hormones, so these for the steroid hormones, they are found in the cytoplasm. For the non-steroid hormones, these are found on the nucleus, like on the nuclear membrane. And that would include things such as thyroid hormone, retinoic acid, vitamin D, etc. And then there are some antagonists of those receptors listed. And then the final thing on this handout, I just want to finish this, and then I want to tell you what we're going to do for Thursday. So there are also this then is what would be the steroid hormone receptors. Found in the cytoplasm. And then here, this is the non-steroid nuclear receptors. So you can see they're sometimes they're on the membrane, but sometimes they're also in the nuclear cytosol. And the hormone will get in through the nuclear pore, and it binds to a inactive receptor heterodimer here. When it's inactive, if you see this here, when these nuclear receptors are inactive, they're bound to a co-repressor. So that means that co-repressor is stopping from having any actions on the DNA in the nucleus. So when the hormone gets through into the nucleus and binds on these already formed dimers, so in this case, these are already dimers. This is distinct from what we've seen with the other 3 receptors we talked about today. And what happens when the hormone binds is it kicks off this co-repressor and recruits again RNA polymerase and a co-activator, just like we saw with the steroid nuclear receptors. This then leads to transcription of mRNA. mRNA is then translated by the ribosomes into proteins, and then you can have altered cell function. Okay, so just to recap, today we started with GABA, which is an ionotropic receptor. We then went to these kinase receptors, start as monomers, then become dimers when they're bound together. Then we went to the cytokine receptors, which also are monomers, so very similar to the kinase receptors, except they do not have any intrinsic enzymatic activity. They are bound to inactive kinases that become active when the dimers form. And then we have nuclear receptors, either those in the cytoplasm of the cell or those on the nuclear membrane or the cytoplasm of the nucleus. And that's going to change the mechanism a little bit. The ones that are the receptors that are in the cytoplasm are monomers, just like we saw with the cytokine and kinase linked receptors, and dimerize upon binding of the ligand. Whereas the non-steroid nuclear receptors that reside in the nucleus or on the nuclear membrane, they are already dimers, right? And so once they are activated, they're dimers that are bound, first to this co-repressor, and once they're activated, that co-repressor is kicked off. But after that, the mechanism is very similar to what we saw with the cytoplasmic nuclear receptors. Okay, so what I want you to do for Thursday is I want you to pick one out of this handout. I want you to pick one of the receptors that you went over, whether it's like one of these G protein coupled receptors, one of the kinase receptors, one of the ion channel receptors, anything. And I want you to find a primary research paper on, or some kind of research paper on a drug that it acts on these receptors. And I want you to, it'd be good if it's like a primary research paper, because it'd be like maybe one of the first times they identified these drugs. And I want you to talk about how that drug is used, like what diseases can it be used to treat, and then talk about its mechanism action as it relates to the receptors that you went through in this handout. And then the mechanism of action, how it helps treat that disease or the disease symptoms. It would be great if you could all kind of talk to each other about which one you're going to pick, because I wouldn't want everyone to pick the same one. So there's 1, 2, 3, 4, 5, 6, 7 of you, and 1, 2, 3, 4, 5, 6 different types of receptors on this page 4 here, right? That we've gone through. Or actually 5, 1, 2, 3, because intracellular enzymes is different. But here, GPCR, ion channels, transmembrane enzymes, transmembrane non-enzymes, and nuclear receptors. So there's 5. So there's going to be some overlap between you because there's 7 of you. But if you talk to each other, make sure that we at least cover these 5 different classes once. So, 5 of you take one of these, and then the other 2 of you can double up, but just make sure you're not picking the same drug bind to these receptors. Does that make sense to everyone?
Yes.
Yes.
Yes? Okay. So if you can find that paper and summarize it, that's what I plan to do on Thursday, is go through what you find out in those papers, okay?
Dr. Kavanaugh, it's better if we decide now, because I think after the class we will be on a different lab and different time. So
okay. So why don't we I'll just go down. All right, I'm just going to go down and assign them then. So Afsana, why don't you do G protein coupled receptors? Melanie, you do ion channel receptors. Matt, you can do transmembrane enzymes. Vivek, you can do transmembrane non-enzyme receptors. Yang, do nuclear receptors.
Sure.
Okay, and then Chetan, we're going to go back to G protein coupled receptors and Muslim ion channels. So Muslim and Melanie, please talk that you don't get the same drug, okay? And Asana and Chetan, can you talk so you don't get the same drug for the G protein coupled receptor?
Okay.
Okay?
Okay.
Okay.
Yeah, makes sense?
Yeah.
Yes.
Okay. Okay, and I thank you for your patience with me today, not having my iPad. I don't know what I was thinking. And also by doing Zoom for the whole meeting. I hope to be able to be on campus on Thursday, probably wearing a mask, but I hope to meet you in person on Thursday in 708 Fisher, correct?
Yes.
Yes.
Yes. Yeah. Okay, and if you have any questions about the assignment between now and then, just let me know. I'll also type it up and put it up on Canvas. Let me write myself a note, make sure I know who everyone has. So this is Afsana and Chetan here. This was Ion Channels was Melanie and who else had Ion? Young was
Muslim.
Yes.
Muslim, thank you. And transmembrane enzymes was you moved around on my thing here, so I just went down the thing. Matt, was that you?
Yes.
Yeah.
Yes, it was.
Yes. And then transmembrane non-enzymes was Vivek.
Yes.
Yep. Okay. And then that's it for class today. I'm going to regroup with Dr. Johnson because just the last time we did this class, I did it on my own and I did it in a little different sequence. So I'll just regroup with him for next week. But for this week, we're just going to have you, that's going to be your homework to present those papers on Thursday. Okay. And so there's you have an hour and a half. We might have to go into next Tuesday a little bit if we go over, but I'm thinking like 10 minutes, like nothing too elaborate. Just give us the general idea of the paper. And if you could send me the paper you're going to use, because I'll put it up on Canvas so everyone can kind of see it. No, everyone can see it. Not kind of see it. I'm sorry. I'm just like. Okay. Okay. Well, I look forward to seeing you all in person on Thursday, and let me know if you have any questions, and I'll post this up on Canvas for you all.
Okay, thank you.
Okay, thank you, everyone.
Thank you.
Thank you.
Thank you.
Bye bye, Dr.
Bye.
Bye-bye.